All right, welcome everybody. Hope you're all enjoying this wonderful conference. Today's session is on emotion, motivation, and social neuroscience. So our first speaker is Yury Koush from Lausanne, who will talk about dynamic brain networks engaged in positive emotion regulation.
Okay, thank you very much for the invitation. I will tell about dynamic brain networks engaged in positive emotion regulation. Oscar Wilde wrote, "I don't want to be at the mercy of my emotions. I want to use them, to enjoy them, and to dominate them." Indeed, in everyday life, we maintain, suppress, or change the nature and the intensity of our emotions. Emotion regulation allows us to adaptively cope with negative and positive events of our daily life, and it constitutes an important aspect of our personal well-being and social interactions. So failure to do so can result in burdening affective disorders. Model of cognitive control of emotion highlights neurosystem involved in using cognitive strategies to regulate emotion, such as reappraisal. For example, it involves neurosystem attributing mental states, conflict monitoring, doing selective attention, and working memory, selection, and inhibition. It involves neurosystem involved in generating emotional responses. So the systems that provide arousal threat value of stimuli, encode reward value of stimuli, and represent associated body states. There are neurosystems with intermediary roles in reappraisal, for example, those that value stimulus in current content and represent perceptual and semantic features. So why positive emotion regulation? We know very well that emotion regulation is mostly addressed using reappraisal and suppression as cognitive strategies to decrease negative emotions, first of all. So engaging in positive emotion is a complementary strategy that can promote recovery during cognitive therapy of mood disorders. Now, it facilitates creative thinking, decision-making, problem-solving, and social bonds. The impairment of positive emotion regulation contributes to the severity of mood disorders, as one example, anhedonic symptoms associated with depression. So our study was designed in a very standard way. So we measured 25 healthy participants in 2x2 factorial design. We used factors stimuli with positive scenes versus neural objects, and we used standard photographs for that. For task, we selected passive viewing task versus imagining to positively experience the same object from the first-person perspective. So we used a few standardized psychometric questionnaires and picture set ratings before and after our study. For analysis, we used whole brain random effect 2 ANOVA. We linked psychometric measures to brain activity, performed meta-analytic functional profiling and functional connectivity modeling, and finally, dynamic causal modeling. For whole brain analysis, we found significant main effect of task and on stimuli. For main effect of task, we found activity in lateral part of the dorsomedial prefrontal cortex. Among others, for main effect of stimuli, we found central part of dorsomedial prefrontal cortex, ventromedial prefrontal cortex, and bilateral amygdala. These regions we analyzed in our subsequent analysis. So now, if you check our clusters. So we have 5 areas here, and we want to see which emotion and cognition and perception eventually categories we can get if we check these clusters with meta-analytical databases. So that's what we are doing here. Of course, as expected, for left, right amygdala and ventromedial prefrontal cortex, we have more likely categories of emotion and perception. But what we see for central part of dorsomedial prefrontal cortex, for example, that there are additional categories such as cognition, social cognition, and for paradigm, the theory of mind. So interestingly, for lateral part of dorsomedial prefrontal cortex, cognition and social cognition category and theory of mind paradigms became more likely. As I mentioned already, we did not see valence and arousal rating differences before and after experiment because our study did not focus on training emotional regulation. However, we saw that higher level of thought suppression predicted lower levels of activity change in the lateral part of dorsal medial prefrontal cortex and the ventromedial prefrontal cortex. Now, what if you look at functional networks? And we use for this seed-based analysis of meta-analytic connectivity model. And what does it mean? That we have one seed, and now in a huge database we look at which area are functionally connected to this particular seed. What's interesting here is that if you take each seed as a separate analysis, we see that resultant co-activation maps are strongly overlap, which speaks in favor of common involvement of these areas in our particular experiment. Now, finally, we have dynamic causal modeling, which is a Bayesian framework to model functional brain networks. So 5 ROIs models result in 2 power 45 model alternatives in total. It was too much. So we use hierarchical DCM analysis to reduce the model space. And we suggested these 22 different model architectures, which differ in how hierarchically the network might behave. So we have our prior hypothesis from cognitive neuroscience background, for example. Now, what we do, we model external stimuli to these models and modulatory inputs. And the whole set of models is estimated for two paradigms, for two conditions, sorry. So we have passive viewing condition for positive pictures and emotion regulation condition for positive pictures as well. So to be more precise, we have a winning model family using the Bayesian model inference that DCM provides us with. So we see here that when you look into the differences on how external stimuli might input our model, we can find that for passive viewing, external stimuli entered ventromedial prefrontal cortex. For emotion regulation, we see that external stimuli enters ventromedial prefrontal cortex and lateral part of dorsal medial one. Here we also see the parameter estimates and how they propagate within the network. So this exploratory analysis of the network allows us to suggest that top-down cognitive control processes originate in the lateral part of the dorsal medial prefrontal cortex, control affective valuation of external input in the ventromedial one, and that emotional response processes originate in the amygdala and modulated via central dorsal medial prefrontal cortex. So now, highly specialized prefrontal cortex area and the amygdala brain network dynamics suggest a common role in cognitive appraisal. To sum up, I would like to say that we have different but complementary analysis, and using them allow us to explore in total, the dynamics and the functionality under our experimental task. Well, thank you very much for your attention. Here's our team.
Thank you. Are there any questions? Just step up to the mic. Maybe I can start. You call your task an emotion regulation task, but as I understood, it's also really a memory recall task, right? You're remembering experiences?
Yes. So the positive social, for example, or just a scene, positive scene, was used as a clue. That means participants were free to associate any positive experience to this particular picture. So there was no limitation. It could be positive memory, of course.
Okay, thank you again.
Yeah.
So our next speaker is Matthew Hutchison from Harvard University. His talk is titled Brain Responses to Free Viewing of Dynamic Social and Non-Social Object Interactions.
Hi, everyone. Most of my research is focused on looking at brain dynamics, which doesn't really make me a social neuroscientist, but hopefully this will still tie in the literature well enough. The reason why we chose to go after social brain networks is a lot of work, really meticulous work, has been done mapping out this really distributed set of regions that make up the social brain. And performing elements of social cognition typically gets fractionated into this distributed hierarchy of perception, mentation, affect, motor regions. Motor's grayed out mainly because I'm not going to be talking about those regions today, but you can see this makes up a large area of cortex that encompasses lower order visual areas all the way up to higher order association cortex. Typically when people are studying these regions, they use very isolated stimuli. Cropped faces or specific text stories that will require subjects to mentalize. These are very unnaturalistic stimuli, but they offer very exquisite control over what's shown to the subjects, and you can make more definitive conclusions based on that. But the world that we actually engage and interact with is much more dynamic. We're moving through an environment that's also moving. We have multiple senses that are being stimulated, and we're interacting with individuals. And because of that, there's been a pitch to go another way, get more complex, have subjects simultaneously in the scanner interacting in like a hyperscanning kind of situation. So there's kind of these two ends of a spectrum. There's actually a nice middle ground using naturalistic movie stimuli. Yury Koush showed this really well in 2004. He had subjects passively view a movie, the good and the bad and the ugly. And then what he did, and you can see in this red trace at the bottom, is he just averaged the time courses of regions of interest, in this case in the fusiform gyrus. And you can use a reverse correlation approach and find when are there peaks in activity and look to see what scenes elicited that activity. So even though these subjects are free to look wherever they want in the movie, the faces are moving in and out of the scene, they're partially occluded by hands, you're still able to robustly elicit responses in the fusiform face area as if you were showing somebody an isolated cropped face. So this was a particularly motivating finding when we designed our test, which involved having subjects watch consecutive 12-second movie clips. And these contained either objects, people interacting with objects, or people interacting with other people in some sort of social dynamic. And we had people do 2 runs where they watched 30 movies in each run. And I should say that they watched this without sound. We scanned 232 subjects, and then from there we created 2 different groups, the discovery and replication group that were matched across age, education, IQ, and signal-to-noise properties of the actual scanning data. And the first thing, obviously, to do is just the standard general linear model, so we can contrast the social scenes where there are people interacting versus the person scenes where there are just people interacting with objects. And we see a very canonical network pattern, similar to some of the regions I showed highlighting, everything that we expected to find in terms of activity along the superior-temporal sulcus or the dorsal medial ventromedial prefrontal cortex, the posterior cingulate cortex along the fusiform gyrus. We find the expected network. So these naturalistic stimuli, these very brief movie clips played with no sound, and essentially you don't have that much context, but you're trying to interpret it, is able to elicit a very robust pattern that is virtually identical between our 2 groups. The downside with this approach is that we don't really know what was going on during the scan. Overall, we see that these areas are more active relative to the person-object scenes. But if we want to explore ongoing cognition and ongoing brain dynamics, we need to look deeper. And we're going to use an approach similar to what Yury Koush did, so looking at perceptual regions. It's a relatively complicated plot that you can look at closer in a poster session later today. But what you're seeing here are 2 traces, one from the discovery group, one from the replication, from the fusiform face area overlaid on the task structure, and then you're seeing just movie frames highlighting certain clips. And you'll see that there are, and this is percent signal change, so that there are scenes that selectively and robustly activate the fusiform face area. You see these large magnitude changes in the signal relative to baseline, and it's specific to person-object scenes and social scenes, so when there are faces in the actual scene. And what's also really exciting about this is we see virtual 100% correlation value of 1 in terms of the timing and magnitude between the 2 groups. And this isn't happening just when faces are present. You can look at traces where there's a green background, so just the object scene. Even though there's a negative dip, the magnitude of how low that goes and the timing of that is identical between the 2 groups. This is the perihippocampal place area or lateral occipital cortex, and we find very specific patterns as would be expected based on isolated stimuli. When we go into higher order areas, which typically haven't been looked at in this approach, we find a very similar finding, very tight coupling between the subjects that are very specific to the content of the movie scenes. The posterior cingulate cortex responding and peaking in all the yellow blocks, so when there are social scenes. And you can start going into these individual scenes and find what elicits activity to a greater degree than other even movie classes of the same content. And you can start dissociating areas. So the posterior cingulate cortex and temporal parietal junction both showed up in the general linear model. But here we can start finding scenes. Can't use a laser pointer here, but if you look at the 4th from the left, the posterior cingulate cortex does not show any change in magnitude, whereas the temporal junction, you see a robust response. And it seems to be the case that posterior cingulate cortex is much more specific to movies that have social scenes in them, whereas temporal junction is responding when, even if there's one person in the scene, if it requires any type of mentalizing to understand the intent of the character. And we can use this as an exploratory approach to start generating hypotheses to test with more in-depth analyses. The anterior superior temporal sulcus follows a very similar trend as the temporal junction. Dorsal medial prefrontal cortex is much more specific in its response, typically responding in scenes that have a lot of conflict between the characters or complex interactions. What's also interesting, and I alluded to it by talking about the cingulate temporal junction dissociation, but typically it's believed that these networks are working coherently all the time. We show people stimuli, we assume all regions are engaged when it's that certain type of stimuli class. So this is looking at single volumes and looking at the activation pattern in that volume. So I'm just plotting the magnitude of that here. And at certain time clips within social scenes, you see that all the regions of interest have a high level, suggesting that they're all functionally connected at that point. But in other clips that are also showing social stimuli, we find that they can get dissociated. In that first instance where there's a child boxing, the posterior cingulate cortex is actually negative relative to the other regions. In other scenes, you'll see the dorsal medial prefrontal cortex go in the opposite direction. So at different time points, we're actually engaging different parts of this subnetwork, which had previously been believed to be really working more coherently. So we can actually start differentiating individual movie or content responses. I don't have a lot of time to go into affect, but we do find affective regions are also driven by these to a lesser degree, which is probably owing to the lower signal-to-noise property in subcortical regions and ventromedial PFC. And not all our areas show robust responses. We actually had seeded the nucleus accumbens, and these were defined a priori. These weren't defined from the general linear model contrast, and we don't really find that the movies elicit any response. So it's not general across all regions. And I've been showing you group maps, so averaged across the 77 subjects, but we can look at this at an individual level. So it's a colorful plot, but what this is all 232 subjects plotted in the y-axis, and this includes actually subjects with autism spectrum disorder. And what you're seeing is the time series of, in this case, the MT complex, color-coded and stacked on top of one another. And what you see is here is a remarkable consistency. So we have a varied age, IQ, demographics, such as race. These all vary, and yet we have very stereotyped responses across the subject. And this is the case for higher order areas as well. You can see, again, the ventromedial PFC is more noisy, but even in something like temporal junction, it's amazingly consistent at an individual subject level as they watch they freely view these movie clips. And you also see slight evidence of some right lateralization here, but again, we see pretty stereotyped responses in both hemispheres. And for me, this is kind of the highlight is we don't even have to choose individual regions. We can run this at an individual voxel level. So what you're seeing here is essentially real-time brain activity. It's been interpolated to 1 second, and this is the movie clips that they're seeing. And there's been no hemodynamic lag introduced in the clip. So they see the movie. If you watch 5 or 6 seconds, you'll see the peak activity that's the result of seeing that content of the movie. And there are coherent patterns that emerge here. So again, this is voxel-wise. And very little pre-processing. Essentially, this is just averaging subject data as they watch the movie. And you can see the brain responses. And there are very stereotyped responses, and there are interesting responses that emerge, but allows us to start characterizing our data more towards the real-time aspect, to actually understand what elements of the brain are working at a given moment in response to a given task, instead of just averaging across the data to create a single picture. Because there are idiosyncrasies, whether it be the luminance or the content in the frames and how they're engaging, that we need to consider. And I'm just going to let this run for one more clip because this is a good social clip. So you see here 2 people engaging, looking at a photo. And if you watch the brain, you're going to see these social regions really come online. Superior temporal sulcus, ventral and dorsal medial PFC, the posterior cingulate. And then as the scene transitions to an object scene, all those areas start to decrease, and you see other sets of regions come online. So it's an interesting approach that, again, will drive hypotheses and further testing. This is consistent between 2 groups. So that was the discovery group. The replication group shows virtually an identical voxel-wise pattern, volume for volume. So the hemodynamic delays are identical between the 2 groups. And we can start clustering. I don't have time to go into it, but we find that certain types of stimuli tend to cluster the types of patterns that emerge over time. So hopefully I've at least convinced you to consider at least this approach in generating hypotheses and examining your data, and that we can reliably revoke stereotyped responses from naturalistic stimuli. Thank you.
Any questions?
Hi. Nice data, Matt. I was wondering, you said you had some ASD subjects there.
Yes.
You want to give us a little teaser of what you saw there?
Yeah, so interestingly, we started with about 50 ASD subjects. These were clinical patients. By the time we cleaned out the motion and took out the subjects that we weren't really happy with, we only were left with a small sample. So we had 20 subjects, but they were very clean data. We saw no difference. Even when we cheated and ran the GLM analysis and chose regions that were close to statistically significant, the peak magnitude in some of the regions, you can find some variability, but on a global scale, we see the exact same stereotyped response in the ASD subjects in all the areas, including higher order regions. So it seems like when we control for motion, they're still these are still functioning enough to show up to the scan, but clinically diagnosed ASD subjects at a region-wise response at a second-to-second level, we see the virtually identical bold response. So it's indicative that maybe imaging might not be the right way to go in addressing this because the macroscopic changes perhaps aren't there. But essentially we're getting as in detail to the signal as we can, and we don't find large differences between those groups. But again, it's a very small subset. 20 subjects isn't really a lot to make definitive conclusions, but it does point more towards the null.
Have you examined the effect of one scene on the next, so like of the social context or of the interactions between different kind of contents?
We haven't done that yet. The one thing I'm sure most of you notice is we actually don't have a baseline condition in the task. So we don't, now we're starting to do more data collection. We scan, we have a fixation scan, so we can't actually estimate a real baseline for each individual frame. So you can't actually contrast them as well because they're predicated on what was shown before. But yeah, and we haven't gone into detail enough of decomposing the scenes into the individual features, like even the overall movement yet, but that's in the works.
Maybe just one last question. Can you correlate other measures such as connectivity measures in real time? Have you tried that?
Correlating?
Using connectivity between regions as the measure that you're correlating across subjects.
Yes, you could absolutely do that. Yeah, and so like Yury and even his original paper, we're using intersubject correlation. People now do dynamic intersubject correlation. So essentially, you're looking at the functional connectivity between your regions and everybody else's regions at the time, and you get a map. And we've also done that. The only downside is you keep getting further and further away from the original signal. And the goal here was to show that you don't need to do a fancy analysis. This is just a mean, and you can start pulling out relevant information about what's going on neurally.
Thank you very much. So our next speaker is Christine Macare from King's College London, who will talk about neurobiological pathway to smoking in adolescence.
Ooh. All right, thanks. So bonjour, mesdames et messieurs. That's as far as my French goes. As I've been introduced, my talk is going to be about adolescence and smoking in adolescence in particular. And I'm taking an imaging genetics approach, so a big chunk of my talk is actually more genetics than imaging, but I'll guide you through it. Guess this way? Oh, all right. Okay, so smoking in adolescence. First of all, you might think, why is it even important to study adolescence? So what I've been showing you here is data from epidemiological studies, particular one from Hans-Ulrich Wittchen, where you can see age plotted on the x-axis and basically the percentage of a drug use, or in this case nicotine use, plotted on the y-axis. And you pretty much see that there's a time window when it all emerges, and this overlays with the start in the time course of adolescence. So most of smokers who actually go on to become adult smokers and nicotine addicted have initiated smoking during adolescence. Now, thinking further, you might want to know what are the factors that drive actually smoking initiation in the first place. And so from behavior genetics approaches, we know that one factor which is important in smoking, in explaining inter-individual differences in smoking is are genetic factors. So in fact, 44% explain inter-individual differences in smoking initiation. Now, going deeper into what these genetic influences actually are and where they are located. Previous work has been done by Jules Gelernter, who was the first one to report the TTC12-ANK1 DRD2 gene cluster and its relevance in smoking. And what he found was strong association with a European American sample. And then a colleague of mine went on further to look at that in a longitudinal perspective. So what she did is she investigated specifically variants in this gene cluster, and she looked at the association with smoking in adolescence as well as mid-adulthood, and she found stronger associations in adolescence. Now, thinking about this gene cluster, so one thing which all of these genes have in common is they can be functionally linked to the brain dopamine system. And I've highlighted here in red the reward system, which is the dopamine system in the brain. Now, one area, one candidate area which is particularly involved in the brain's dopamine system is the striatum, in particular the ventral striatum, as Nora Volkov has been, for instance, talking about in her keynote yesterday. And so we thought in terms of characterizing the neurobiology that underlies the pathway from the TTC12-ANK1 DRD2 gene cluster to smoking in adolescence would be to look at striatum activity and the ventral striatum in particular. So here are our aims for this study. So we first wanted to identify SNPs within the gene cluster that are actually associated with self-reported cigarette smoking in adolescence, and we also validated our self-report with plasma cortinin levels. We then went on to investigate the gene expression of these functional SNPs or of the relevant SNPs in terms of their expression in post-mortal human brain tissue. And then we looked at the relevant SNPs and their genotype effects on reward processing in adolescence by looking at the bold signal in the ventral striatum during reward processing. Just quickly to give you an overview of the samples, or the samples that we used. So we had a total sample size of 14,000 individuals, and these were derived from 4 different adolescent cohorts, mainly the Northern Finland birth cohorts, 2 of them, as well as the Alsbach cohort, which is the AVEN longitudinal study located in Bristol, and the Imogen cohort, which is an EU-wide adolescent longitudinal sample. So as you can see, all of these are quite comparable in terms of their ages, and they all contributed to our total sample size. Now, one thing to realize is that smoking was assessed in different ways in these different samples, so we needed a way of concatenating the data most efficiently. And so we came up with 4 categories wherein we broadly categorized participants. And so never tried were those individuals who have literally never reported never having tried cigarette smoking. Ever tried were the ones who reported having tried at least once. And out of this group, there were people who reported having smoked on more than one occasion or more than one cigarette. And we characterized them loosely as smokers. And the ones who reported regular patterns in smoking, so for instance, over the last couple of months, we characterized them as weekly smokers. Now, one thing to be aware of with self-reported smoking is that it is a proxy of nicotine intake, and in adolescents in particular, it's important to have validation of self-report, as that's due to reporting biases. So we used a subset of data from the Alsbach sample, which had plasma cortinin levels, and cortinin is a metabolite which is derived from nicotine. And what you can see here is that we have a validation, basically, of our self-reported smoking. Those participants who actually reported never having smoked, indeed did have very, very low levels of mean cotinine levels. So we can basically trust our self-report. So after we've established that, we went on to look at the genetic association in the whole sample, and we used as a reference group the participants who reported never having tried smoking, and we compared them to the ones who report having tried smoking, to smokers as well as to weekly smokers. And what you can see here is the locus which we identified in our first comparison in the TTC12 gene, which we then went on to investigate in the smokers and the weekly smokers, and we obviously replicated that in there. Now, then we went on to investigate the risk variant, or the risk allele within of the SNP, which is the minor allele, the G allele, and we looked at the risk with smoking in each of the samples. So what you see here are forest plots, again for ever tried smokers and weekly smokers. And the big black boxes basically reflect the size of the study that went into. And what you can see here is that we find associations across all comparisons. And these were all significant. We then went on to investigate gene expression, as we wanted to functionally characterize that SNP that we found. And so we used a publicly available database for that, which contains post-mortem brain tissue, and we focused specifically on striatal tissue, as gene expression differs per tissue type in the brain. And we wanted to focus on the reward system and chose the striatum as a candidate for that. And so what you can see here in the first graph, in graph A, is basically developmental stage plotted on the x-axis versus expression levels log transformed on the y-axis. And you can see that TTC12 and ANC1 are very weakly expressed throughout the lifetime, whereas DRD2 is more strongly expressed in the striatum. And it appears to show an increase, or at least a peak, during adolescence. We then looked at the gene expression pattern of DRD2 and associated that with our risk allele, our G alleles, and found a significant association there. So basically, the higher the number of G risk alleles, the more your DRD2 expression in the striatum. It's probably important to mention that this is in a relatively small sample, but nevertheless important data. Last but not least, we investigated reward processing during the fMRI task, and those of you who have heard about the Imogen study or familiar with it know that that's a European population sample of 2,000 adolescents, and they've been very well characterized at the ages of 14, 16, and 19, and so this is data from age 14. And this is one of our fMRI tasks called the monetary incentive delay task. I'm not sure if you're familiar with this task. I'm just going to briefly explain it. So basically, participants are presented with three conditions: a no win, a small win, and a big win condition. And the task has different phases. So there's an anticipation and a feedback phase. Here, I'm going to focus on reward anticipation. So that would be the first bit of the task. Basically, I'm going to look at the contrast data comparing brain activation or the bold signal during no win versus that during the big win in the big win condition. This is basically the whole brain map which we get from the data, and from this we extracted ventral striatum, the region of interest based on previously reported coordinates, and when we associated that brain activity with our risk allele. And what we did here, since this sample was the sample of 1,000 roughly adolescents, wherein the sample sizes were quite low in terms of the G allele carriers. So we pooled all G allele carriers and compared those to the AA homozygotes. And we found a significant association with the bold signal in the left ventral striatum. Let me just point out that we found one in the right ventral striatum as well, but that one was didn't survive correction. Okay, so to sum up, in terms of our aims, we first wanted to investigate SNPs within the TTC12 and ANG1-DID2 gene cluster, which were associated with self-reported smoking. In adolescence, we found one locus where the G allele carriers showed a higher risk at for smoking initiation. We validated our self-reported cigarette smoking in adolescence by plasma cotinine levels. We looked at gene expression in the striatum in post-mortem human brain tissue and found an association with our SNP, in particular our G allele there, our risk allele there. And then we were thinking about how to interpret this result because our SNP is located in TTC12. And we thought this SNP might potentially focus as an eSNP, which affects expression in genes which are located closely to the actual gene where it's coming from. And so there might be potential interaction effects there. And then last, we looked at an association between the bold response in the striatum, in the ventral striatum in this case, and our G allele carriers, and found a significant association there as well. So that's pretty much it. I'd like to thank my collaborators, and I thank you for your attention.
Thank you. Yes, go ahead, question.
Thank you. I want to know whether did you collect other genes such as MAO?
We did not look at other genes in this particular study, but the possibility is there to look at data from other genes. Obviously, we wanted to focus on this this gene cluster because it was previously reported to be important in smoking, but there's obviously a possibility to look at other genes there as well.
And did you perform the craving in the middle elder and adolescent?
Sorry, say that again?
A craving in the adolescent and the middle elder. Have you measured that?
No, we didn't. We didn't look at craving. No. So we wanted to focus here on smoking initiation, since these people were quite young, so they were adolescents. They did not actually have that much time to develop nicotine dependence and craving as such. So we really wanted to focus on the ones who initiate smoking early on versus the ones who don't.
Thank you.
Thanks a lot.
Okay, thank you very much.
Thanks.
So our next speaker is Daniel Shaw from Brno, Czech Republic, the dynamic brain during interaction.
Okay, hi everyone. I'm going to skim past some of the details here. And if some of it isn't clear, I just don't want to run out of time, but if it isn't clear, I urge you to come to our poster, which we'll be presenting immediately afterwards. Okay, so in the Behavior and Social Neuroscience group, in which I'm a part, we've become increasingly interested and focused on understanding the neural dynamics, the neural processes, mechanisms that afford our ability to co-adapt with our interaction partners almost seemingly and almost very smoothly. And to understand that, we have combined our two identical 3 Tesla scanners into this hyperscanning protocols, and it's allowed us to look at, by varying the paradigms, to really tease apart the co-adaptation that is occurring between individuals while they're actually interacting. This slide is perhaps slightly redundant now because my intention was to really emphasize the necessity of hyperscanning to look at these kind of processes in the brain, but as it seems to me quite obvious that the community is really coming around to the potential benefits of hyperscanning to look at these kind of processes. So it's I don't need to oversell that, I don't think, to you guys. So to look at co-adaptation, within the hyperscanning setting, we utilized the Ultimatum Game, which is a really simple paradigm where a proposer is offered a sum of money, a division of a sum of money, and he chooses which of two divisions to offer to a responder. And the responder's choice, quite simply, is to either accept that division or reject it. If he accepts it, the money is divided accordingly. If he rejects it, then no of the players make any money. And it's usually played as a single-shot game, so just one pair playing one economic exchange, and then the behavior of multiple pairs are aggregated. And neuroeconomic and social neuroscientific research to date has really focused almost exclusively on neural responses of the responder during this game, because it shows that responders can behave quite irrationally in the sense that they will reject offers that they consider to be unfair, even though any money is better than zero. And the neural processes, the neural reactions or responses within the responder's brain show that these almost irrational behaviors are underlined or associated with, driven by effective responses in the bilateral insula and the anterior cingulate cortex, but also in the inferior parietal lobule and in the dorsolateral prefrontal cortices. But we wanted to capture the co-adaptation that might be emerging during social interaction on this task. We used an iterated ultimatum game where we had 19 male-male dyads who performed 120 rounds of the ultimatum game within the scanner. And the idea here was that in order to maximize the potential gains to yourself, you really need to co-adapt with your interaction partner as the game unfolds. So a very simple schematic to show the experimental layout. It was performed in an event-related fashion, and we had 120 of these experimental trials. Now, we had 60 of two different kinds each of trial types. The first trial type was a PR condition, which is I'm showing you now. That's where either the proposer or the responder walks away with a maximum relative gain. Or there was a PP condition where the proposer is going to walk away with a maximum relative gain regardless of the two choices. It's just a matter of how selfish he wants to be at that time. And then there's a very simple control condition which was intended to eliminate any low-level visual or motor processes. And there were 60 control trials, okay? So 60 trials of each type. A very quick and crude snapshot of the behavioral measure. I'm just looking here at the total number of 12 of offers and corresponding acceptances for the most unfair offer. I'm saying unfair. That's the offer that presents maximal advantageous inequity, where the proposer is making the most and the responder is consequently making the least, the most unfair offer. And we have choice sets on the left here that were within the PP condition, and those on the right in the PR condition, showing greater number of unfair offers on the PR condition. But what's important, there are two main things I want to take away from this. The first is that the offers, the unfair offers are presented on the bottom here. I can't point to them, but they're presented on the bottom. And the number of unfair offers are not a function of the absolute gain that the proposer is taking away from it. There's something else going on. And we can also take the difference in the log ratio of the choices to show the inequity present within the choices. And we can show actually the inequity in the choices also isn't driving the proposer's responses. Something else is going on. And we think that what was going on is that the proposer was essentially adapting to the behavior of the responder and reacting to them. And we can capture this with the Cox model, which I don't have time to go over in detail. But essentially, the predicted utility from a given choice is taken as a function of the gain to the individual, the gain to the other individual, weighted by the emotional state of the individual making the choice. The emotional state within the Cox model is broken down into two parameters. We focused on one, which is reciprocity. That basically reflects the degree to which I am more likely to value your gains, irrespective of my own, if I feel that you have treated me kind in the past, or you will treat me kind in the future. Likewise, there's negative reciprocity, where I'm more likely to devalue your gain if I think that you've done me harm in the past, or you will do me harm in the future. So we can quite simply incorporate this to model the expected utilities. So every individual has then a reciprocity parameter, a specific reciprocity parameter. That's quite easy to model then the responder's behavior using that reciprocity parameter. For the proposer, we went one step further and we said they will choose, they will not only have reciprocity in their own, but they will be trying to predict the reciprocity of their responder. And they will choose the fair offer if they consider the reciprocity of their responder will be in some way beneficial in the future to themselves. So we're trying to capture the reactiveness, the responsiveness, co-adaptation of the two players to one another. When we model the expected utility for the proposer using these reciprocity parameters, we find that a given choice set isn't the predicted utility of a given choice set isn't static, it's unfolding over time in response to their inferences of the responder's reciprocity. When we plug that in as a behavioral modulator at the individual level, what we see in the responder at the top, we see, yes, the same neural systems that are responding to unfair offers in the responder. We can capture that by modeling their behavior with this reciprocity parameter. But what's more striking, perhaps, is that the proposer, there seems to be having some kind of mirror-like response in these emotion-driven or emotion-related centers in the insular cortices, the anterior cingulate. And we thought perhaps what's going on is that there is some kind of empathic response going on whilst they're offering these unfair offers. Perhaps they're hypothesizing how the other person might be feeling to this. So now we can capture the true essence, the sexiness of hyperscanning data, and we can look at correlations between the two interacting players. So what we do for each voxel, in a voxel-wise manner, we take a spatially corresponding voxel in both players' brain. We extract the time series throughout the whole time course. We identify all the trials that are related to each of the three conditions, and we concatenate the time courses according to each condition. So each player has a time course for each pair has a time course for each, a correlation time course for each condition. We then take, for the 19 dyads, we have 19 whole brain volumes of correlations, which we convert to Z-scores. Then we can perform paired samples, random effects t-tests to assess differences in the degree of intra-dyad, or interplayer if you like, but I'm saying intra-dyad to emphasize the fact that it's within the pairs, the degree of correlations between them. And what we find, looking now at the PR greater than control condition and the two experimental conditions compared, we find that the degree of intra-dyad correlation is greater on the experimental trials, and this appears within the anterior cingulate, within the bilateral anterior insula, and in these areas that are popping out when we're looking only at the responder's brain responses to unfair offers. This gives for us some support to the idea that the proposers are engaging in some sort of empathic response. And finally, what we can do is we can look at the intra-dyad correlations within the right anterior insula. That's the area that showed these strong correlations in the experimental comparison. And we can see that there's a positive relationship with the number of unfair offers that they are making on this particular trial. We can also take a measure of co-adaptation, a crude measure, but it's just simply the number of unfair offers that were accepted. And we plug that in as a behavioral regressor at the group level to look at the contrast between PR and control. And we see that that measure of co-adaptation, or at least similarity and fairness judgments, predicts the intersubject correlation within the anterior cingulate. So this is bringing together the two interacting brains and relating it to their behavioral performance. That's everything from me. Big thanks to all the people that helped to make this possible.
Are there any questions? You have to go to the mic if you want to ask.
There's no
question,
hiding.
Please.
Just a quick question. Were the participants, did they know each other? Oh, I'm in the middle.
Okay.
Did they know each other previously, or were they complete strangers?
They were absolute complete strangers, and we made sure that there was no familiarity whatsoever. We controlled also, because the two scanners are located in essentially adjacent rooms, it meant that we could have real control over that aspect. Mm-hmm. That was really important for us.
would you expect there to be a difference in your patterns of activation if they were known to one another?
Absolutely, yeah. That's one of the things that we're looking at now, using a slightly different paradigm, but trying to evaluate the degree that familiarity will really influence this. We would expect so, of course.
Go ahead.
All right, my turn now. I was wondering whether you've acquired personality data on that data set as well, and whether you've looked at associations with certain personality features, psychopathy.
We don't have personality data, but what we do have, which I think will be quite exciting, is we have some performance measures such of, we've developed, I say we, that's the royal we, I didn't, but our group developed the Czech version of the multifaceted empathy test, a performance measure of empathy. And we also have measures of self-other distinction and things like this. So we don't have personality, but we have some other performance-based measures that maybe will be related to intra-dyad correlations.
All right.
Definitely something to consider there.
Okay, yeah, thanks.
Okay, thanks again. So our next speaker is Chong-Eun Han from Montreal Neurological Institute, McGill University.
So hi everyone. So today I'm going to talk about our study that looked at how a hunger hormone ghrelin affects food odor conditioning in humans. So every day we are bombarded by attractive food cues, and this may contribute to the obesity epidemic that we are facing today. Indeed, food cues have been shown to have incentive value and trigger an urge to eat, non-homeostatic eating, and ultimately weight gain. But these cues are not naturally salient. They are initially neutral stimuli, but after being repeatedly paired with food, they become predictive of food delivery. They gain incentive salience, and they can induce these feeding-related behaviors. One system that subserves this reward cue-associative learning is the dopamine system. And dopamine modulates the learning by generating signals that reflect reward prediction error, which is the discrepancy between predicted and experienced reward. So during each and every food cue exposure, dopamine computes reward prediction error and updates a value that is assigned to food cues, potentially affecting feeding-related behaviors. There is a hormone called Ghrelin, which is shown to increase dopamine signaling that occurs during food cue exposure. But ghrelin is actually a homeostatic signal that reflects energy balance. It is produced mainly from the stomach and acts on the hypothalamus and the brainstem to trigger hunger and feeding. But ghrelin has its receptors also on dopamine neurons of the ventral tegmental area. So ghrelin injection directly into the ventral tegmental area can trigger dopamine release, and it can also increase dopaminergic activity and perhaps influence the processes that are subserved by the dopamine systems, which is the reward cue learning as well as valuation of food cues. Indeed, now, there is a lot of evidence from both animal and human studies that show that ghrelin promotes incentive value of food cues, perhaps via the dopamine system. So, for example, in our lab, we've shown in humans that ghrelin injection is associated with increased activity in the brain regions that are related to incentive salience in response to food pictures, and also with better recall of these food cues. But what's unknown in humans is whether or not ghrelin also participates in food cue associative learning. So this is the question that we wanted to explore in our study. And we did so by injecting people with ghrelin and observing how fMRI responses during food odor conditioning are affected by high levels of ghrelin. More specifically, we recruited young, healthy men and women whose body mass index were within the normal range. We asked them to come to our lab for 2 fMRI sessions following ghrelin or saline injection. So when they first came to our lab, we gave them a standard breakfast. 3 hours after breakfast, which was when ghrelin levels are supposedly at the lowest level, we injected them with ghrelin or saline, and we placed them into the scanner, during which they performed this associative learning task. So during this task, they learned to associate abstract images with food or non-food odors or airless odor. So 2 images were paired with food odors, 2 images were paired with non-food odors, and the other 2 were paired with air. And also, in order to induce reward prediction error, the images that predicted odor delivery were followed by odors only 50% of the time, and the other half of the time, the expected odor were omitted. So as you can see in this figure, each trial began with image presentation. 500 milliseconds after, its corresponding odor appeared, and both stimuli disappeared at the same time at 1.25 seconds. For each image, subjects were also instructed to indicate whether or not the images were made of straight or curvy lines, and some subjects also rated pleasantness of the images that they saw throughout the scan. 24 hours after, they came back to our lab to again rate the pleasantness of the images that they saw during the scan the day before. So we tested our hypothesis, which was that ghrelin promotes learning that is specific to food stimuli, and we did so in 3 different ways. First, we looked at hedonic ratings of the abstract images. So as the associative learning occurs, the images should gain incentive value. And if ghrelin promotes food associative learning, then it should be that the images paired with food odors be rated more pleasantly following conditioning during ghrelin versus saline condition. We also looked at reaction time, which is shown in some studies to reflect learning and also be negatively correlated with value. And based on our hypothesis, we predicted that subjects will react faster on food trials in the ghrelin condition. In order to look at the neural correlates of learning, we computed signals that reflect learning, which are expected value and reward prediction error using a reinforcement learning model. So according to this model, the reward prediction error, or RPE, is basically the difference between the actually received reward value and expected value. And the expected value was calculated by adding the value of a previous trial to the prediction error multiplied by the learning rate that we calculated using our reaction time data. And we correlated our brain data, our brain activity with the expected value at the time of image presentation and also with reward prediction error at the time of odor delivery. And we predicted that these RP-related and value-related activity would be greater during food conditioning following ghrelin treatment. So before running the main analysis that tested our hypothesis, we had to make sure that odor perception occurred to induce learning. And fortunately, we were able to see that odor processing was associated with increased activity in the brain regions that are previously known to subserve dysfunctions, such as the prefrontal cortex and the orbitofrontal cortex. So now the main results that tested our hypothesis. So in terms of reaction time, we saw decrease in reaction time over the runs in both ghrelin and saline conditions, regardless of the event type, which may indicate that learning had occurred. We also saw a significant interaction between treatment type and event type, such that following ghrelin treatment, reaction time was faster on food trials compared to non-food trials and air trials. Our hedonic rating results were also consistent with our hypothesis, such that after a 24-hour delay, the images that predicted food odor delivery following ghrelin treatment were rated to be more pleasant compared to the images that they never saw before. So now the fMRI data that we correlated with expected values at the time of image presentation and also with reward prediction error at the time of odor delivery. So we saw that expected values correlated with activity in many brain regions in both ghrelin and saline conditions that included the amygdala, the prefrontal cortex, and also the insula. But we tested our hypothesis specifically on the regions that are previously shown to be related to subjective value according to a meta-analysis that was conducted by Bartra and colleagues. And in one of the clusters that includes the ventral medial prefrontal cortex, we saw that value-related activity during food conditioning was greater in the ghrelin compared to saline condition. In another cluster that is associated with subjective value that largely includes the nucleus accumbens, we also saw significant value-related activity during food conditioning, but this did not differ between ghrelin and saline conditions. But interestingly, we saw that this value-related activity correlated significantly with pleasantness ratings that were collected during the scanner, but only following ghrelin treatment. Another index of learning, reward prediction error, correlated positively with many similar brain regions, including the amygdala, piriform cortex, and also striatal regions, including the ventral striatum. Similar to what we did for the value-related activity, we tested our hypothesis only on the clusters of brain regions that are previously identified to be associated with reward prediction error, according to another meta-analysis, and we observed that a significant interaction between treatment condition and event type, such that following ghrelin administration, the reward prediction error-related activity was greater on food trials compared to non-food trials, and this RP-related activity correlated significantly with in-scanner pleasantness ratings only following ghrelin treatment and only during food conditioning. So to summarize, we tested our hypothesis that ghrelin promotes learning that is specific to food reward, perhaps via stimulating the dopamine system. And our results appear to support our hypothesis, as we've shown that ghrelin increased reward prediction error-related activity in the relevant brain regions only during food conditioning. And potentially as a consequence, the incentive value that food-related stimuli gained appeared to be stronger following ghrelin injection, as we've shown that ghrelin increased value-related activity in the ventral medial prefrontal cortex, and it also increased pleasantness of the images that predicted food odor delivery. So ghrelin, by acting on brain regions that subserve motivation and learning, may have a role that is beyond homeostatic eating. Thank you.
Are there any questions? I can't ask a question because it's my work as well, so no one has any questions? Okay. Okay, go ahead.
Here.
Thank you.
One question here.
Okay.
Did you also analyze the ghrelin level in the blood?
What we looked at was the levels of growth hormone. So the receptor that ghrelin binds to in the brain actually stimulates growth hormone levels. So what we measured was the level of growth hormone in the blood. So we made sure that ghrelin increased dramatically the levels of growth hormone. Yeah.
Question here.
When you showed the association between the nucleus accumbens and the pleasantness rating, I'm not entirely sure whether I missed something, but the sample size seemed to have dropped for that?
Yeah. So only... Yeah.
What happened there?
Yeah, so we collected our data in sort of two different batches. So in the second set of subjects, we asked them to rate the pleasantness of the images. That's why it's only a subset of subjects that rated those things.
Okay,
yeah.
Thanks.
Okay, thank you very much.
Thanks.
And now our final speaker is Laura Cuaya from the Institute of Neurobiology in Querétaro, Mexico. Interspecies face processing in dogs.
Hi. Hi. In this meeting of the Organization for Human Brain Mapping, ironically, I will talk to you about dog's brain. So we will need a new logo just for this talk. Yes. Now it's perfect. But the short question in this talk is, what happens in the dog's brain when they see a human face? But maybe your first question is, why should we study the cognition of another species? And the answer is because we use another species in the world. Also, when we study the cognition of another species, we are learning about our own cognition. And the cognitive ethology can be summarized by this beautiful phrase by the professor Mark Bekoff. studying animal mice and hares. And now, thanks to the fMRI technique, we can also study their brains. This is amazing. But now, why dogs? And the one reason is because dogs are an excellent model to study social cognition because of their perfect abilities to interpret humans. As a species, dogs are evolutionarily far from humans, but from thousands of years, dogs and humans have shared their environment. But more inspiring reason was exposed by Adam Microsi. He said, if we have the challenge to understand the mind of another species, why should we not choose our best friends for this endeavor? After of reading this inspiring phrase of a review, I decided to study dogs in my PhD. And the first question was about the abilities, the human face perception in dogs. Because a human face is a really important thing to dogs. With just a glance, dogs can determine the gender, the identity, and even the attentional and emotional state of a human. And now, the most important aspect, the welfare of our participants. Well, all participants are pets from local families, and they come to the lab only in special days. The rest of the time, they live with their families. During all sessions, dogs are awake and unrestrained, and they can decide to leave the session at any moment. Yes. Well, here I'm showing the steps of the training. In A, we teach to the dogs to maintain the Sphinx position. In B, when the dogs was able to maintain this position by 5 minutes, they come to the lab and we teach maintain the same position, but now inside of a scanner replica. In C, they explore the environment of the real scanner, and finally in D, they are maintaining the Sphinx position inside of a real scanner, and they are wearing protection against the scanner noises. But the truth is that not all the sessions are perfect. Above, I'm showing the parameters, and we use a block design with 2 types of blocks, human faces and objects. The total duration of each run was almost 3 minutes, and each dog had 5 runs in total. And now, the first result, and a dog's brain. Here, a dog's brain. The contrast was human faces versus objects, and we found 2 clusters. One located in the left temporal cortex and extend to the frontal cortex, thalamus, as well caudate. And the second was located in the right frontal cortex and extend to the temporal cortex. The activity in the temporal cortex is good because in other species, the temporal cortex is also related to the perception of faces. But the activity in the frontal cortex and the caudate is more interesting, but also more difficult to interpret, because we didn't if this activity is because human faces or is for faces in general. So, to try to get to the bottom of this, we did another experiment. And the only difference is that now we use dog faces like stimuli. And this is the result. We found a cluster in the temporal cortex, and this cluster was extended to the occipital cortex and cerebellum. And all the activity is in the left hemisphere. Using all this data, we did a more integral analysis, and here the result. This is the result. First, we did a conjunction analysis for all visual stimuli. In other words, what cerebral regions share the processing of objects, dog faces, and human faces. This is in blue, and this is the occipital cortex in both hemispheres. We believe that this result is consistent with the role of the occipital cortex as visual area. Now, the second conjunction analysis is present in red. And this is the more important to the question about the face perception in dogs. Before, we saw that the contrast faces versus objects show a lot of areas related to the face processing. But with this analysis, we found that the only cerebral regions that process facing is the temporal cortex. This is the temporal cortex in dogs is related to processing faces of humans and dogs. This is a great result because in humans, no humans, primates, sheep, and now dogs, temporal cortex is related with this processing. And the more interesting result is in purple. This is compare the two contrasts: human faces versus objects against the human faces versus objects. And we found that the activity in the frontal cortex is related only to human faces, not to dog faces, neither objects. So we believe that this means that the frontal cortex is the cerebral correlates of the behavioral patterns that dogs show only to human faces. And finally, the almost poetic result. We found that the caudate in dogs is related only to human faces. We use stimuli faces of strangers, and another team has vinculated the caudate with processing of rewards. So it is possible that dogs interpret a human face, even of a stranger, like a reward. So we show that dogs are dogs. They love humans. Here, our participants, the entire project was possible because of their cooperation, and it is me and my dog, Kun Kun. My highest hope is that this kind of projects make a difference in the way that humans interact and interpret dogs. I believe we were more aware about the cognitive and emotional life of dogs, then we improve their life around of the world. Thanks.
Thank you so much. This is so amazing presentation. I really love it. And I'm unable to ask questions because I'm so excited. I have just a suggestion. Do you want to continue this line of research by using, of course, what I'm talking about, point-light biological motion stimuli? Because human motion and probably dog point-light biological motion of dogs would be also an important part of social cognition. And if so, I would love to cooperate with you.
Thank you. It is a great proposal.
There's a question over there.
Yes, thank you. That was a very, very interesting talk, entertaining talk. I'm wondering, so in terms of, have you also then been looking at dogs with strangers versus their owners' pictures? And then also, do you have a dog social reciprocity scale? So, because different dog breeds and things are more social than others, and are you thinking about looking at some kind of dog scale of social reciprocity and how it relates to these findings as well?
Yes, these breeds of dogs is the more intelligent. This is cheating, and we use these dogs because they have a high degree of motivation. And the faces of owner is our dream. Yes.
Thank you.
Thank you.
Okay, thank you very much. Thank you, everyone, for attending.